Chapter 5 Genomics, Proteomics and Bioinformatics. Genomics Introduction and Concept. Genomics is the scientific disciplines of mapping, sequencing, and analyzing the genome. And the genome is the basic set of chromosomes or the complete set of chromosomal and extra chromosomal genes of an organism. We know that the organism that contain the cells, that contain the nucleus, and within the nucleus there is the chromosomes. And the total set of the chromosomes, complete set of the chromosomal and extra chromosomal genes of an organism, that is called as the genome. So the genome is simply the total number of the chromosomes or the total set of the chromosomes along with the entire genes and the nucleotide sequence as such. Genomics can be defined as the study of molecular organization of genome, their information, content, and the gene product they encode. It means that what type of the genes that encode the particular type of the characters of that genes. And that character that is responsible for the particular gene that can be directly encoded by the help of the gene and the total information and the content of the gene product that they encode. It is the rapid and accurate method of the gene mapping sequencing means the determining the exact order of the bases in the strand of DNA. We know the DNA that is the double-stranded DNA molecules which contain the particular bases and the phosphate and sugars. So that can be determined by the help of the entire that process, and that is called as the sequencing, that is called as the gene mapping, and this is nothing but the entire that what we used to say the genomics. The genome sequencing provides the knowledge of the total number of genes and the relationship between the gene. It can give the idea regarding the total number of the genes and the relationship between that genes, what type of the relationship that has been established within the genes. However, they enter any type of the organism that has such concern that contain the particular set of the chromosomes and what type of the entire that genes which are located on the entire that chromosomes and what type of the relationship that can be directly determined. And this determined by the help of the Winkler, especially the Winkler coined the term genome in 1922. The first genome to be sequenced was the microbe Haemophilus influenzae in 1995. Then other organisms like E. coli, Saccharomyces cerevisiae in 1997 and the human genome in 2003. The type of genomics. There are three types of the genomics: structural genomics, functional genomics, and the comparative genomics. Let us see one by one. The structural genomics. It is the study of the structure of entire genome of living organisms. It means that what type of the set of the chromosomes are there, what type of the set of the entire the genes are there, that can be studied by the help of the structure. And this is nothing but the structural genomics, living organism, which determine the size of the genome of specific in the megabases. It determines the first structure of the proteins and then function. In every organism, about 12 to 15% of genes are structural genes. Functional genes, second, it is the study of the function of all genes present in the entire genome. All set of the chromosomes, all set of the entire the genomes are the structural chromosomes, functional chromosomes, what chromosomes are present within them, and along with that chromosomes, what type of the genomes are there, total set of the chromosome, total set of the genes that can be directly determined, and their expression. It deals with the transcriptions and proteomes. The transcription means the set of the RNAs transcribes from the genome, and the proteomes refer the complete set of the protein encoded by that genome. What type of the proteins are there that can be encoded by the help of particular type of the genome, and that is nothing but the proteomes. Third one, that is the comparative genomics. It deals with the predictions of the genes and the function of these genes by the comparing different genomes. What type of the entire different genomes are there and how it can be predicted with the help of these genes. So these predictions of the genes and the function of the genes by comparing their different genomes. In this type, the whole part of the genome resulting from the genome projected and project are compared to the study the basic biological similarities and the differences as well as the evolutionary relationship between them. Methods used for the whole genome sequencing. What type of the methods are there that can be directly sequences that have been made? How the genome sequencing is made, and there are methods, and these methods are following. DNA sequencing is the process of identifying the order of nucleotide present in DNA sequence. What type of the nucleotide sequence are present? Whether it may be the adenine, cytosine, thymine, adenine, guanine, the thymine, cytosine. What type of the entire sequence are there? Nucleotide sequence are there. These DNA sequencing is a process that can identify the order of nucleotide present within the DNA. Following are the methods of whole genome sequencing. First, that is the dideoxy chain termination method. It is also called as the Sanger method because it is developed by the help of the Sanger in 1975. The principle of this method is that incorporation of 2-3-DDNP, dideoxy DNTP, in the place of its corresponding DNTP in DNA chain terminate the DNA synthesis because the DDNT pairs nucleotide analogous with the LAC 3-hydroxyl group, that is the OH group of necessary of phosphodiester bond formation and the chain elongation. In this method, the single-stranded DNA used and synthesized in vitro in 4 separate tubes, and each tube contains the different DDNP. For example, DDNP NTP in tube 1. Hence, the chain termination will occur wherever the adenine to be incorporated in it. At the end of the reaction, the series of DNA fragments of the different lengths are produced in each tube. One radioactive deoxynucleotide is also incorporated. This is incorporated for the encoding, incorporated into the tube. Then samples are run on the sequencing gel. The bands are seen from the bases. That is, the gel electrophoresis apparatus may be utilized in this method. The advantages of this method: first, the long reading sequence up to 900 BP base pair. Second, easy to analyze and high accuracy. Third, relatively small data storage is required. However, some disadvantages are also there. The first disadvantage is low sensitivity, high cost per basis, large amount of startup material is required. Second method is Maxam Gilbert method. This method was developed by Alan Maxam and Walter Gilbert in 1976 to 78. This method is based on the nucleobases. The specific partial chemical modification of DNA and subsequent cleavage of DNA backbone at the site adjacent to the modified nucleotides. In this method, DNA fragments to be sequenced is labeled either 5' or 3' end and divided equally into the 4 tubes. In each tube, chemical reaction is formed to modify one base at a time, and then piperidine reagent is used, which causes the loss of methylated bases and breaks the DNA at a site of lost base. Then the products are separated by the gel electrophoresis and detected by the autotoradography. The advantages of this method, that is the Albert Gin's methods: purified DNA can be read directly. Fragments of DNA can be sequenced. It can be used to analyze the DNA. However, some disadvantages are also there. Not widely utilized. It needs the relatively complex setup, and there is a limitation of gel electrophoresis up to 500 to 700 BP, that is the base pair. Next method, that is the shotgun sequencing. It is the technique for determining the sequence of entire chromosomes and the entire genome based on producing random fragments of DNA. It produces the random fragments of DNA, not exactly the set of DNA, or not exactly the cut pieces of the DNA that can be formed, but it is the random fragments of DNA that can be formed by the help of the shotgun sequencing, which are then assembled by computers, and then that are fragments order the fragments by finding the overlapping ends. This is followed by the four stages, that is the library construction. It is nothing but the isolated and the purified fragments of chromosomes are attached to the plasmid vector like E. coli. Then random sequencing, DNA is purified from the plasmid. The whole genome is sequenced many times using universal primer and with automated sequencer. And third one, that is the fragment alignment and the gap closure. By computer program, the random sequence DNA fragments are clustered and assembled into the longer fragments. It means that there is a cluster formation of the DNA, and that forms the long fiber of the DNA or longer fragment that has been formed, and these long fragments that can be utilized for the separation. Fragments continues and then align to form the complete genome without gap. Fourth, proofreading. Sequences are edited and proofreading is done carefully. Advantages: it is the faster process due to the removal of the mapping stages. Second, that is, it uses the small amount of DNA. However, disadvantages is there. That is costly due to the software and computing power, and it can lead to the errors, etc. Now, second, that is the proteomics. Proteomics is the large-scale study of the proteomes, which is total set of the protein expressed from the transcription of cell. The transcriptome is the complement of mRNA, that is the messenger RNA, transcript from the enter the genome of cell. The proteomics is the identification, analysis, and large-scale characterizations of the proteome expressed by any given cell, tissue, or the organ under defined condition, and the investigation of the proteins when and where they express. The goals of the proteomics are: first, the characterize post-transcriptional modification of proteins. Second, to prepare the three-dimensional map of the cell indicating the exact location of the protein. And proteomics can be provide the significant biological information to many problems. The global analysis of the gene expression protein level is also called as the proteomics. The types of the proteomics: basically, there are three types of the proteins. First, proteomics, expression proteomics. Second, structural proteomics, and third one, that is the functional proteomics. Expression proteomics, that is nothing but it includes the analysis of the protein expression at large scale. It is the study of the expression of the protein between the sample which differ by the some variable quantitatively, such as the diseased and healthy tissue. Third, next, that is the structural proteomics. Second, it is the study of the structure and the nature of the protein complex present particular organelles which characterize the protein-protein interaction. What type of the interaction in between the two proteins are there, that can be directly determined. Third one, that is the functional proteomics. It is the use of the proteomics methods for analysis of the properties of molecular network formed in the living cell. It includes the isolation of the proteins complex. Methods used in the proteomes analysis. The traditional methods for the proteome analysis are ion exchange chromatography, size exclusion chromatography, and affinity chromatography. For selective protein, the enzymes linked immunosorbent assay, and for the complex protein, sample sodium dodecyl sulfate polyacramide gel electrophoresis apparatus is utilized. Two-dimensional gel electrophoresis and the two-dimensional differential gel electrophoresis are used. For quantitative proteomics, many techniques like the isotopes coded affinity tag labeling, stable isotopes labeling with amino acid in the cell culture are developed recently. It provides the information about the significance of the proteomics. Now, the significance of the proteomics. The study of the proteomics is necessary because protein govern the phenotypic characters of the cell. Here we should know that what are the characters of the phenotypic characters as well as the genotypic characters. Genotypic character that directly refers the entire the genome or whatever the entire the genetic makeup of that organism, that cell, and the phenotypic characters that directly determine how that genome and how that entered the cells, how that organisms eats. Second, it provides the information about the genome function. What type of the entered function of the genomes are there, it can directly determine, and the information that can directly provided by the help of such type of the proteomics. Now, third one, that is the proteomics helps in the identification functional domains in the the protein sequences. What type of the protein sequences are there? transcription, that is the mRNA or the proteomics are there, that can be directly determined by the help of the proteomics. Next, that is the proteomics is important for early disease diagnosis. Proteomics that can directly determine the early disease of the diagnosis. Whatever the early stage of the particular type of the diseases in the human beings are there, that can be directly determined by the help of the proteomics. It means that the proteomics that can directly analyze the protein-protein relationship, that can directly analyze the entire the protein-protein relationship within the entire cells, and that directly provide the information regarding the what type of the early stage of the entire the diseases are there, that can be directly determined by the help of the proteomics. And fifth, that is, it is the vital role in the drug development as well as the target molecule. Next, that is the bioinformatics. Bioinformatics, that is this is such a so-called as a information technology, and this information technology that can directly have the data in the database, whatever the informations are there, that is in the form of the database, and this database that can be utilized, that is the bioinformatics as such. The introduction and concept. It is the information technology applied to the management and the analysis of the biological data. What type of the biological data of that organisms are there, that can be directly stored in the database, and this database that can be utilized as a tools for the bioinformatics as such. It is also defined as the applications of the tools of computations and analyze to interpret the biological data. It can directly interpret the biological data and analyze that biological data. It is an interdisciplinary field and computer science. Interdisciplinary field and computer science. Mathematics, physics, biology comes under it. The bioinformatics is the important for the management of the data in modern biology and the medicine. Database and its classification. Database is the computerized and organized storehouse of related information which provide a way for searching the searching, inserting and updating the data. It is like a library, consists of the database. The database can be classified into the 4 categories on the basis of information stored. What type of the information is stored and this how it is supposed to be stored in the, what type of the database, and that is why it is into 4 groups. There follows the primary database. In this type of database data, experimentally derived data is stored, so called archival database. What type of the experiment that is done? If the experiment is finished and whatever the results and the conclusion that can be directly stored in the enter this type of the database. That is the primary database. It stores the information related to the sequence of the structure of biological component. Sequence database is applicable to the nucleic acid, and the protein sequence and the structure database is the only proteins. Once database is the accession number is given, the data in primary database is never changes and it become the archival scientific record. It becomes the scientific record. The most common primary databases are the DNA Databank of Japan and the European Molecular Biology Database or European Molecular Biology Laboratory, that is EMBL. Secondary database. It contains the data obtained through the analysis of data from the primary database. Whatever they enter the database that has been obtained from the primary database, that is the archival database, that is stored database, and these database it obtained and analyzed this information that is analyzed, and these analyzed database that is nothing but the secondary database. Third one, that is the composite database. These are the collection of the many primary databases resources with primary and secondary characteristics. It means that the first, second that can be directly included, that is the composite database. NCBI, that is the National Center for the Biotechnology Information, it is the most important database, that is the composite database, is a composite database which stores the sequence of nucleotide and the protein. And fourth one, that is the specialized database. They provide the particular research interest, that is the HIV sequence database, whatever it may be. Example of databases. First, GeneBank. It is the popular nucleic acid sequence database of NCBI, freely available for the public. GeneBank. It is comprehensive database which contains more than 240,000 nucleic sequences available to public. Each GeneBank entry include concise description of the sequence, scientific names and the taxonomy of the source organism. Second one, that is the EMBL, that is the European Molecular Biology Laboratory. These EMBL nucleotide sequences database is a comprehensive database of DNA and the RNA sequence. Submitted researcher and the genome sequence group and collected from the scientific literature and the patent application. EMBL database library was established in 1980 to collect, organize and distribute the database nucleotide sequence data. And third one, that is the UniProt, Universal Protein Resources. It provides the staple, comprehensive, freely accessible resource of the protein sequence and the functional annotation. It is a collaboration between EBI, Protein Information Resource, Swiss Institute of Bioinformatics. Manual storage of the protein sequence by the computer analysis. Their archiving and the development of the website are carried out. SwissProt, Flybase are the example of the database. Importance of the databases. First, database act as a storehouse of the information. This is very clear. So whatever they enter the database, that data is stored, and this database that can be utilized for the researcher. This database can be utilized by the help of the scientist also. It is an asset as a retrieval of variety of the search criteria. Third one, it acts as a reference library for the molecular biology. Yes, it is right. An example, the secondary database which provide the information, for example, gene or the gene product has been investigated by the research community. Fourth one, that is the data retrieval tools. Use of the database requires some research engine, use of the database requires some search engine and analysis tools. The entries of the database needs to be retrieved in the magnificial format is done by these tools. Following are the DRTS, that is the data retrieval tools. Entries. It is the integrated database system of NCBI and cover over 20 databases. It can be used to access literature, prepare genome maps, nucleotide and the protein sequence, and get the structural data. Second, OMIN, online Mendelian inheritance in man. This database was initiated by Dr. Victor A.M. as a catalog of Mendelian traits and the disorder. It is non-sequence based information source useful in genomics. Third one, that is the PUBmed. It is the free full text archives of the biomedical and the life science journal literature, which provide the more than 11 million journals citation of MEDLINE, that is Medline, that is the medical library analysis and retrieval system online. Fourth one, that is the taxonomic browser. It is a synthetic database that allows the users to examine the progress of DNA barcoding by browsing the different levels of the taxonomic hierarchy. Application of the bioinformatics. First, gene prediction can be done by using the bioinformatics tools. Second, protein sequence can be reduced from the predicted genes using the computer programs. And third one, that is the regularly sequence can also be identified and analyzed.